Understanding and manipulating the gut microbiome for personalized medicine. The gut microbiome is the collection of microorganisms that inhabit the human gastrointestinal tract. It is estimated that the gut microbiome contains more than 100 trillion bacteria, as well as viruses, fungi, archaea, and protozoa. The gut microbiome plays a crucial role in human health, as it influences various aspects of digestion, metabolism, immunity, and brain function. The composition of the gut microbiota is shaped by multiple factors, such as genetics, diet, lifestyle, environment, and medication. However, the gut microbiome is also dynamic and adaptable, and can be altered by various interventions. The study of the gut microbiome has been greatly facilitated by the development of advanced techniques that allow the characterization of microbial communities at different levels. Metagenomics is the analysis of the collective genome of the gut microbiota, which reveals the diversity and functional potential of the microbial population. Metatranscriptomics is the analysis of the gene expression of the gut microbiota, which reflects the metabolic activity and response to environmental stimuli. Metabolomics is the analysis of the small molecules produced by the gut microbiota, which represent the end products and intermediates of microbial metabolism. These techniques rely on next-generation sequencing and bioinformatics tools to generate and interpret large amounts of data. The gut microbiome has been implicated in various health conditions, both within and beyond the gastrointestinal tract. For example, obesity has been associated with changes in the gut microbiota composition and function, such as reduced bacterial diversity, increased abundance of firmicutes and decreased abundance of bacteroidetes, and altered production of short-chain fatty acids. Inflammatory bowel disease, IBD, which includes Crohn's disease and ulcerative colitis, has been linked to dysbiosis of the gut microbiota, characterized by reduced bacterial diversity, increased abundance of proteobacteria and decreased abundance of firmicutes and bacteroidetes, and altered immune response to microbial antigens. Allergies have been suggested to result from an imbalance between regulatory and effector T cells in the gut mucosa, which may be influenced by the gut microbiota composition and function. Mental health disorders, such as depression and anxiety, have been shown to be associated with changes in the gut microbiota composition and function, such as reduced bacterial diversity, increased abundance of Enterobacteriaceae and decreased abundance of Lactobacillus and Bifidobacterium, and altered production of neurotransmitters and neuroactive metabolites. Personalized medicine is an emerging approach that aims to tailor healthcare to each individual's unique characteristics, such as genetics, environment, lifestyle, and preferences. The gut microbiome can be considered as a key factor in personalized medicine, as it reflects both intrinsic and extrinsic factors that affect human health. The gut microbiome can be used for personalized diagnostics, therapeutics, and interventions. For example, the gut microbiome can be used as a biomarker to diagnose diseases or predict responses to treatments. The gut microbiome can also be used as a target to modulate diseases or enhance health outcomes. The gut microbiome can also be used as a tool to deliver drugs or genes to specific sites in the gastrointestinal tract. The manipulation of the gut microbiome can be achieved by various strategies that aim to modify its composition or function. Dietary interventions are one of the most common and effective ways to modulate the gut microbiota, as different types of foods can selectively stimulate or inhibit certain microbial groups or metabolic pathways. Probiotics are live microorganisms that confer health benefits to the host when administered in adequate amounts. Probiotics can modulate the gut microbiota by competing with pathogens, producing antimicrobial substances, enhancing mucosal barrier function, modulating immune response, and influencing brain activity. Prebiotics are non-digestible food components that selectively stimulate the growth or activity of beneficial microorganisms in the gut. Prebiotics can modulate the gut microbiota by increasing bacterial diversity, producing short-chain fatty acids, and influencing immune response and brain activity. Antibiotics are drugs that kill or inhibit bacteria. Antibiotics can modulate the gut microbiota by reducing bacterial diversity, altering bacterial composition, and affecting bacterial function. However, antibiotics can also cause adverse effects on the host, such as diarrhea, resistance, and dysbiosis. Fecal microbiota transplantation, FMT, is a procedure that involves transferring fecal material from a healthy donor to a recipient with a disease associated with dysbiosis. FMT can modulate the gut microbiota by restoring bacterial diversity, replacing missing or depleted bacteria, and correcting bacterial function. FMT has been shown to be effective in treating recurrent Clostridium difficile infection, and is being explored for other conditions, such as IBD, obesity, and mental health disorders. The manipulation of the gut microbiome poses several challenges and ethical considerations that need to be addressed before its widespread application in personalized medicine. Some of these challenges include identifying optimal donors or probiotic strains for FMT or probiotic therapy, standardizing protocols for FMT or probiotic therapy, monitoring safety and efficacy of FMT or probiotic therapy, understanding long-term effects of FMT or probiotic therapy, assessing individual variability in response to FMT or probiotic therapy, integrating multi-omics data for comprehensive analysis of the gut microbiome, developing reliable biomarkers for diagnosis or prognosis based on the gut microbiome, designing personalized interventions based on the gut microbiome, ensuring informed consent and privacy protection for donors or recipients of FMT or probiotic therapy, addressing ethical issues related to ownership, accessibility, and commercialization of FMT or probiotic therapy. The field of gut microbiome research is rapidly evolving and expanding, offering new opportunities and challenges for personalized medicine. Some of the future directions and research frontiers include elucidating the mechanisms underlying microbial metabolites and their effects on host physiology and pathology, investigating the interactions between the gut microbiome and other host systems, such as the immune system, the nervous system, the endocrine system, and the epigenetic system, applying synthetic biology to engineer novel microorganisms or consortia that can perform specific functions or deliver therapeutic agents in the gastrointestinal tract, evaluating the impact of the gut microbiome on precision medicine and public health, and developing novel tools and platforms to monitor and manipulate the gut microbiome in real time. Innovative therapies targeting the gut microbiome. The gut microbiome is the collection of microorganisms that inhabit the human gastrointestinal tract. It plays a crucial role in human health and disease, as it influences various aspects of digestion, metabolism, immunity, and brain function. The gut microbiome is also dynamic and adaptable, and can be altered by various factors, such as diet, lifestyle, environment, medication, and infection. Therefore, modulating the gut microbiome has emerged as a promising strategy for developing novel therapies for various conditions. Fecal microbiota transplantation, FMT, is a procedure that involves transferring fecal material from a healthy donor to a recipient with a disease associated with dysbiosis, or an imbalance in the gut microbiota. FMT has been used for centuries in traditional medicine, and has gained renewed interest in recent years due to its remarkable efficacy in treating recurrent Clostridium difficile infection, CDI, a potentially life-threatening condition caused by antibiotic-resistant bacteria. FMT has also shown promising results in treating inflammatory bowel disease, IBD, such as Crohn's disease and ulcerative colitis, by restoring the diversity and function of the gut microbiota. Ongoing research is exploring the applications of FMT in other conditions, such as obesity, diabetes, irritable bowel syndrome, and neurological disorders. Future developments may include optimizing the selection of donors and recipients, standardizing the protocols and delivery methods, and developing synthetic or defined fecal material. Probiotics are live microorganisms that confer health benefits to the host when administered in adequate amounts. Probiotics can modulate the gut microbiota by competing with pathogens, producing antimicrobial substances, enhancing mucosal barrier function, modulating immune response, and influencing brain activity. Probiotics have been used for various conditions, such as diarrhea, constipation, allergies, and depression. However, the evidence supporting the use of specific probiotic strains is often inconsistent or inconclusive, and depends on several factors, such as the dose, the duration, the formulation, and the individual variability. Synbiotics are combinations of probiotics with prebiotics, which are non-digestible food components that selectively stimulate the growth or activity of beneficial microorganisms in the gut. Synbiotics can enhance the therapeutic effects of probiotics by increasing their survival, colonization, and function in the gastrointestinal tract. Postbiotics are non-viable microbial products or metabolites that have biological activity in the host. Postbiotics include various substances, such as peptides, polysaccharides, lipids, nucleic acids, and small molecules. One of the most studied postbiotics are short-chain fatty acids, SCFAs, which are produced by the fermentation of dietary fibers by certain gut bacteria. SCFAs have multiple effects on host physiology and immune response, such as providing energy for intestinal cells, regulating intestinal motility and permeability, modulating inflammation and oxidative stress, influencing appetite and glucose homeostasis, and affecting gene expression and epigenetic modifications. Postbiotics and microbial metabolites have therapeutic potential in treating conditions such as inflammatory disorders, metabolic diseases, and cancer. Engineered microbes and synthetic biology are emerging technologies that aim to create novel microorganisms or consortia that can perform specific functions or deliver therapeutic agents in the gastrointestinal tract. Engineered microbes and synthetic biology can be used to modulate the gut microbiome by introducing new genes or pathways into existing microbes, creating new microbes from scratch, or designing microbial communities with desired properties. Engineered microbes and synthetic biology can be used for various purposes, such as degrading toxins or drugs, producing vitamins or hormones, stimulating or inhibiting immune cells, and targeting tumors or pathogens. However, engineered microbes and synthetic biology also pose several challenges and ethical considerations, such as ensuring safety and stability of engineered microbes, preventing horizontal gene transfer or environmental release of engineered microbes, and addressing social and legal issues related to ownership and regulation of engineered microbes. The field of gut microbiome-based therapies is rapidly evolving and expanding, offering new opportunities and challenges for clinical applications. Several clinical trials and translational research are underway to evaluate the safety and efficacy of these therapies in various conditions. Potential future applications include using gut microbiome-based therapies for cancer treatment, neurological disorders, and personalized medicine. However, there are also several challenges that need to be addressed before these therapies can be integrated into clinical practice. These include developing reliable biomarkers for diagnosis or prognosis based on the gut microbiome, designing personalized interventions based on individuals' gut microbiome profile, ensuring informed consent and privacy protection for donors or recipients of FMT or probiotic therapy, addressing ethical issues related to ownership, accessibility, and commercialization of FMT or probiotic therapy, and establishing regulatory guidelines and standards for FMT or probiotic therapy. Dietary strategies and nutritional interventions for modulating gut microbiota. The gut microbiome is the collection of microorganisms that inhabit the human gastrointestinal tract. It plays a crucial role in human health, as it influences various aspects of digestion, metabolism, immunity, and brain function. The gut microbiome is also dynamic and adaptable, and can be altered by various factors, such as diet, lifestyle, environment, medication, and infection. Therefore, modulating the gut microbiome through dietary strategies and nutritional interventions can be a promising approach for preventing or treating various diseases. Macronutrients are the main components of the diet that provide energy and essential nutrients to the body. They include carbohydrates, proteins, and fats. Different macronutrients can have different effects on the gut microbiota composition and diversity. Carbohydrates are the main source of energy for most gut bacteria, especially those that ferment complex carbohydrates into short-chain fatty acids, SCFAs, which have beneficial effects on host physiology and immune response. Dietary fiber, resistant starch, and other prebiotics can promote the growth of beneficial bacteria, such as Bifidobacterium and Lactobacillus, and increase the production of SCFAs. Proteins are essential for building and repairing tissues and enzymes in the body. However, excessive protein intake can also affect the gut microbiota negatively, as some proteins can be fermented by certain bacteria into harmful metabolites, such as ammonia, amines, and sulfides. These metabolites can cause inflammation, oxidative stress, and intestinal permeability. Fats are important for cell membrane structure and hormone synthesis in the body. However, high-fat diets can also alter the gut microbiota composition and function, as they can reduce bacterial diversity, increase the abundance of firmicutes and decrease the abundance of bacteroidetes, and decrease the production of SCFAs. These changes can lead to obesity, insulin resistance, and inflammation. Functional foods are foods that have health benefits beyond the basic nutritional value. They often contain phytochemicals, which are bioactive compounds derived from plants that can modulate the gut microbiome. Phytochemicals can affect the gut microbiota by influencing their growth, adhesion, and metabolism, by interacting with their cell surface receptors or signaling molecules, and by modulating their gene expression or epigenetic modifications. Some examples of phytochemicals that have shown beneficial effects on the gut microbiota include polyphenols, flavonoids, glucosinolates, and curcumin. These phytochemicals can be found in various foods and beverages, such as fruits, vegetables, tea, coffee, wine, spices, and herbs. Fermented foods are foods that have undergone microbial fermentation, which is a process that converts sugars or starches into organic acids or alcohol. Fermented foods can promote a healthy gut microbiome by introducing beneficial bacteria or probiotics into the gut, by producing antimicrobial substances or bacteriocin that inhibit pathogens, by enhancing mucosal barrier function and immune response, and by producing bioactive metabolites or postbiotics that affect host physiology and pathology. Some examples of fermented foods that can be included in the diet are yogurt, kefir, cheese, sauerkraut, kimchi, miso, tempeh, and kombucha. The Western diet is characterized by high intake of refined sugars, saturated fats, and processed foods, and low intake of fiber, fruits, and vegetables. The Western diet has detrimental effects on the gut microbiome, as it can reduce bacterial diversity, alter bacterial composition, decrease bacterial function, and induce gut dysbiosis. Gut dysbiosis is an imbalance in the gut microbiota that is associated with various chronic diseases, such as obesity, metabolic syndrome, type 2 diabetes, cardiovascular disease, inflammatory bowel disease, and colorectal cancer. Therefore, dietary modifications are essential for restoring a healthy gut microbiota. Some dietary recommendations include increasing intake of fiber-rich foods, reducing intake of sugar-sweetened beverages, limiting intake of red meat, choosing lean sources of protein, replacing saturated fats with unsaturated fats, and incorporating fermented foods into the diet. Personalized nutrition is an emerging concept that aims to tailor dietary interventions to each individual's unique characteristics, such as genetics, environment, lifestyle, and preferences. The gut microbiome can be considered as a key factor in personalized nutrition, as it reflects both intrinsic and extrinsic factors that affect human health. The gut microbiome can be used for personalized diagnostics, therapeutics, and interventions. For example, the gut microbiome can be used as a biomarker to diagnose diseases or predict responses to treatments. The gut microbiome can also be used as a target to modulate diseases or enhance health outcomes. The gut microbiome can also be used as a tool to deliver drugs or genes to specific sites in the gastrointestinal tract. Emerging research is exploring the interaction between host genetics, diet, and the gut microbiota, and how this interaction can influence individual variability in response to dietary interventions. The potential of precision nutrition is to tailor dietary interventions based on an individual's gut microbiome profile, and to monitor, evaluate, and adjust these interventions accordingly. Neurological and psychiatric implications of the gut-brain axis. The gut-brain axis is the complex network of interactions between the gastrointestinal tract and the central nervous system. It is essential for maintaining overall health, as it regulates various physiological and psychological processes, such as digestion, metabolism, immunity, cognition, emotion, and behavior. The gut-brain axis communicates through neural, endocrine, and immune pathways, involving the enteric nervous system, the vagus nerve, the hypothalamic pituitary adrenal axis, and the gut-associated lymphoid tissue. A key component of the gut-brain axis is the gut microbiome, which is the collection of microorganisms that inhabit the human gastrointestinal tract. The gut microbiome influences the gut-brain axis by producing various metabolites and molecules that can modulate neural activity and inflammation. The gut microbiome plays a crucial role in early brain development and its impact on neural circuitry. The gut microbiome is established during birth and infancy, and undergoes dynamic changes in response to environmental factors, such as diet, stress, infection, and medication. The gut microbiome can affect brain development by modulating gene expression, neurogenesis, synaptogenesis, myelination, and blood-brain barrier function. The gut microbiome can also affect brain development by influencing neuroendocrine and immune systems, which can alter neurotrophic factors, cytokines, and hormones. Disruptions in the gut microbiome during critical periods may increase the risk of neurodevelopmental disorders, such as autism spectrum disorders, ASD, attention deficit hyperactivity disorder, ADHD, schizophrenia, and mood disorders. The gut microbiome is also associated with mood disorders such as depression and anxiety. Alterations in the gut microbiota composition and diversity have been observed in patients with major depressive disorder, MDD, and generalized anxiety disorder, GAD, compared to healthy controls. The gut microbiome may influence mood and behavior by producing neurotransmitters and neuromodulators, such as serotonin, dopamine, gamma-aminobutyric acid, GABA, and acetylcholine, which can affect synaptic transmission and plasticity in the brain. The gut microbiome may also influence mood and behavior by producing microbial metabolites, such as short-chain fatty acids, SCFAs, tryptophan derivatives, and bile acids, which can modulate neuroinflammation, oxidative stress, and the hypothalamic pituitary adrenal axis. Preclinical and clinical evidence supports the role of the gut microbiome in influencing behavior and emotional well-being. For example, animal studies have shown that germ-free mice or mice with antibiotic-induced dysbiosis exhibit altered anxiety-like and depressive-like behaviors. Human studies have shown that probiotic supplementation or fecal microbiota transplantation can improve mood and cognitive function in patients with MDD or GAD. The gut microbiome may also have a potential influence on neurodegenerative disorders like Alzheimer's disease, AD, and Parkinson's disease, PD. These disorders are characterized by progressive loss of neurons and synapses in specific brain regions, and are associated with chronic inflammation and oxidative stress. Gut dysbiosis may contribute to neurodegeneration by altering the production of SCFAs, which can modulate microglial activation and neuroinflammation, by altering the production of bile acids, which can modulate mitochondrial function and oxidative stress, and by altering the production of lipopolysaccharides, LPS, which can induce systemic inflammation and blood-brain barrier disruption. Emerging research suggests that the gut microbiome may be a potential target for therapeutic interventions in neurodegenerative diseases. For example, animal studies have shown that probiotic supplementation or fecal microbiota transplantation can attenuate cognitive impairment and motor dysfunction in models of AD or PD. Human studies have shown that alterations in the gut microbiota composition and function are associated with clinical features and biomarkers of AD or PD. The gut microbiome has also been implicated in ASD, which is a neurodevelopmental disorder characterized by impaired social communication and restricted and repetitive behaviors. Evidence from animal and human studies suggests that gut dysbiosis may be involved in ASD pathogenesis and symptoms. Gut dysbiosis may contribute to ASD by altering the production of SCFAs, which can modulate synaptic plasticity and social behavior, by altering the production of tryptophan derivatives, which can modulate serotonin signaling and mood, by altering the production of propionic acid, which can induce neuroinflammation and oxidative stress, and by altering the production of LPS, which can induce systemic inflammation and immune dysregulation. Gut microbiome-based interventions have been explored as a potential therapeutic approach for individuals with ASD. For example, animal studies have shown that probiotic supplementation or fecal microbiota transplantation can improve social behavior and cognitive function in models of ASD. Human studies have shown that probiotic supplementation or dietary modification can improve gastrointestinal symptoms and behavioral outcomes in children with ASD. The field of gut-brain axis research is rapidly evolving and expanding, offering new insights into neurological and psychiatric health. Emerging therapeutic strategies targeting the gut microbiome may aim to improve neurological and psychiatric health by modulating microbial composition or function, by enhancing beneficial bacteria or metabolites, by inhibiting harmful bacteria or metabolites, or by delivering drugs or genes to specific sites in the gastrointestinal tract. However, there are also several challenges and opportunities in translating gut microbiome research into clinical applications. These include developing reliable biomarkers for diagnosis or prognosis based on the gut microbiome, designing personalized interventions based on individuals' gut microbiome profile, ensuring safety and efficacy of gut microbiome-based therapies, addressing ethical issues related to ownership, accessibility, and commercialization of gut microbiome-based therapies, and establishing regulatory guidelines and standards for gut microbiome-based therapies.
This night is cold in the kingdom. I can feel you fade away. From the kitchen to the bathroom sinking, your steps keep me awake. Don't cut me down, throw me out, leave me here to waste. I once was a man with dignity and grace. Now I'm slipping through the cracks of your cold embrace. So please, please, could you find a way to let me down slowly? A little sympathy, I hope you can show me. down slowly, let me down, down, let me down, down, let me down.